Hi all, this is Dr. Mamta Java, Microbiology faculty DAMS. Now this video module is going to focus on the biochemical reactions in microbiology, which all of you should know from the point of view of your second prof exams, right? So I'm going to take up each biochemical reaction in a separate video, and in this particular video module we're going to focus on the indole test, right? Now indole test is a very commonly done test for identifying certain members of the Enterobacteriaceae family. Now Enterobacteriaceae family is a very important topic for your theory as well as viva, right? Entero means enteric tract, Bacteriaceae means bacteria. So basically this particular family has got bacteria which have got something to do with the intestine. Either they are commensals of the gut, or they cause disease of the gut like diarrhea, dysentery, or they jump from the gut into the bloodstream and then they go here and there and cause disturbance, meaning diseases here and there like meningitis, septicemia, UTI and all. Right now, for identification of all these important members of the Enterobacteriaceae family, we have got to our rescue a set of biochemical reactions, right? You must have heard about the acronym IMVIC reactions. That actually stands for 4 important biochemical reactions: the indole test, the methyl red test, the Vaux-Prosker reaction, and the citrate utilization test, or simply called as the citrate test. Okay, so in this video module I have picked up the very first test of the IMVIC acronym, that is the indole test. Right now, let us understand what exactly is the idea, is the purpose behind doing the indole test, and what's the principle.
Hello, today we are going to discuss gram staining. It's a differential staining first discovered by Hans Christian Grams in 1884. We'll see the procedure. The materials needed is slides, a grease-free, scratch-free, clean slides, marker pens, reagents. What are the reagents we needed? Methyl violet, grams iodine, acetone and diluted carbon fusion. Then filter paper, gloves, discarding jar and compound light microscope. We'll see the procedure. Step 1, primary stain. Primary stain is crystal violet. We need to flood the slide with the crystal violet and wait for 1 minute. At the end of this primary stain, all the organism will be purple color.
Coming to enriched media. Now look at this cute kiddo who is happy with the pizza. So these are the fussy kids who are not happy, or the fussy bacteria who are not happy with the simple or basal media. You have to please them, you have to bribe them a little more by adding something extra to your culture media. In other words, you are converting your dal chawal, the simple cuisine, to an exotic cuisine. For example, this is Italian. I have put up pizza. Okay, so examples of these enriched media. Okay, of course you're not, examiner won't show you this in your viva voce examination, okay, the left one, but the examiner can show you the middle one and the right one, meaning the chocolatey middle circular plate is the chocolate agar, precisely called so because of its chocolatey color, and on the extreme right the bright red medium is the blood agar.
Hello, today we are going to discuss acid fast staining, the procedures. It was first discovered by Paul Ehrlich. After that, it was modified by Zeal Nielsen. We call it as hot method. And the cold method is Kinyun's method. So now we are going to see hot method, Zeal Nielsen method procedure. For that we need the requirements: slides, marker pen, reagents, 0.1% methylene blue, carbon fusion, strong carbon fusion, and 25% sulfuric acid. Apart from that, we have filter paper, gloves, discarding jar which contains phenol as a disinfectant, and compound light microscope. We'll go to the procedure. So we need a heat fixed smear in the staining rack. So after keeping the heat fixed smear, we are going for staining procedure called primary staining. In the primary staining, we are going to cover the smear with strong carbon fusion and allow to stain it for 5 minutes. In the 5 minutes, very crucial, we need to do intermittent heating with the help of a torch below the slide. Should careful, it should not be charred, intermittent. Then when the fume comes, we'll remove. Again we need to repeat. This will happen for the 3 to 5 minutes. 2 to 3 times we need to do the intermittent heating.
Memorizing tip about the meat extract is you can memorize the constituents of meat extract by looking at these two pictures. Okay, what have I put up? Gypsy like beef. Who is a gypsy? Gypsy refers to somebody who is wandering around. So खाना बदोश लोगों को gypsies बोलते हैं. Gypsies are wanderers, and remember they like beef. Okay. Now what is the correlation between this sentence and our meat extract? Remember, gypsies like beef. Meat extract is obtained from beef, and gypsy, the spelling of